Okay, hello everyone. I'm Ryo Nagao from Okayama University in Japan. This paper is my recent article of Nature Communications. The title is Structure of a Tetrameric Photosystem 1 from a Glaucophyte alga Cyanophora paradoxa. The authors are shown here. This presentation is no figures and tables because of copyright of Nature Communications. However, Nature Communications is an open access journal, so I may be able to use the figures and tables in this paper, but I didn't use them in this presentation. If you hope to see the figures and tables in this paper, please download this PDF file. Okay, let's get started. Okay, so the summary of this presentation is the elucidation of the tetrameric PS1 structure from a primitive eukaryote Cyanophora. The background is shown here. Cyanophora is a photosynthetic eukaryote and an evolutionary intermediate between cyanobacteria and photosynthetic eukaryote. Cyanobacterial PS1 is mainly trimer or tetramer, whereas photosynthetic eukaryote have monomeric PS1 cores. However, Watanabe et al. Showed oligomeric PS1 cores in Cyanophora. From this background, we solved the overall structure of Cyanophora PS1 by cryo-EM single particle analysis. Okay, this is the main figure of this presentation. We solved Cyanophora PS1 tetramer structure by cryo-EM single particle analysis at a resolution of 3.8 angstrom. The PS1 monomeric unit is colored green, magenta, cyan, and orange. However, the tetrameric PS1 structure from the cyanobacterium Anabenes PCC 7120 is shown here. So PS1 monomeric unit is colored green, magenta, cyan, and orange. So you can see the different interactions among PS1 monomeric unit between the two species, so Cyanophora and Anabena. So this is the first result of the oligomeric PS1 structure from photosynthetic eukaryote. Okay, next is why such unique interactions occur in Cyanophora PS1 tetramer, totally different from Anabena PS1 tetramer. So the characteristic subunit are shown here. Blue is PSAL and red is PSAK. So you can see different interactions between the two subunits in Cyanophora PS1 tetramer and Anabena PS1 tetramer. In Cyanophora, two PSAL subunits are associated with two PSAK subunits. However, in Anabena PS1 tetramer, there is no interactions of PSAK at the center of PS1 tetramer. Alternatively, two PSAL subunits interact between each PS1 monomeric unit, like this and like this. Other interesting point is the locking of two PSAK subunits in Cyanophora PS1 tetramer. PS1 monomeric unit does not have a PSAK subunit at the corresponding position, and also this PS1 monomer does not have PSAK subunit at the corresponding position. So this is likely due to the structural crash at these positions if the PSAK binding in this PS1 monomer and this PS1 monomer. Next is time-resolved fluorescence spectra in figure 6. So please see the PDF file of figure 6. So there are a lot of fluorescence band from 0 to 4.6 nanosecond in this figure. The most characteristic band is 17 nanometer band, fluorescence band. This band remains until 130 picosecond. This fluorescence property is significantly different from Anabena PS1 trimers, so this indicates excitation energy dynamics differ between Cyanophora and Anabena PS1 tetramer. This is the last figure of this paper. So this is a schematic model of evolution of PS1 cores. About cyanobacteria PS1 trimers, so mutation of PSAL and PSAK may induce the formation of cyanophora types of PS1 tetramer. On the other hand, about cyanobacterial PS1 tetramer, mutation of PSAL and PSAK may also induce the formation of cyanophora types of PS1 tetramer. About eukaryotic PS1 monomer, once cyanophora PS1 tetramer is dissociated into a PS1 monomer, then the cyanophora PS1 monomer bind to other small subunit of PS1 and LHC subunit. So finally the formation of eukaryotic types of PS1 LHC1 supercomplex. So this is a PS1 monomer. Okay, that's all. So thank you for your attention.